name is Graham Chedd from the Alan Alda Center for Communicating Science at Stony Brook University, and this is another episode of Science on Tap. This evening, my guest is Chris Vakoc from an associated institution with Stony Brook University, Cold Spring Harbor Laboratory. He is a specialist in cancer research, and he is using, like thousands and thousands of other scientists, other biomedical scientists these days, a revolutionary new technique of messing around, editing, improving, changing our genes. Not literally yet in us, but in a lot of cells and systems that are going to teach us a lot about us. His specialty is cancer, and we're going to be getting to that a little bit later on. But first of all, this technology is called CRISPR, but it stands as an acronym for a very ugly phrase. Could you tell us what that is?
So it's clustered, regularly interspaced, short palindromic repeats.
Just tripped right off the tongue, right? Thank goodness they came up with CRISPR. So I was hoping that CRISPR, just yesterday the Nobel Prize for the biological sciences, which is technically medicine or physiology, was announced. And I was hoping it was going to be for CRISPR. But it wasn't. It was for another very important discovery, the way that our cells get rid of junk. But why would you think that CRISPR is likely to get the Nobel Prize one day?
Well, in short, it's a powerful tool that's allowing researchers around the world to change the letters of DNA effectively at will in a way that's much more efficient than any technology we've ever had before. If you consider that much of biology is about studying how genes influence biology, this tool is allowing us to manipulate genes in the laboratory with unparalleled power.
Is it really true that most labs now are using this technology?
I think most labs that study genetics, that study how genes work, are using this in all aspects of biology research, not just cancer biology.
And it just came along, what, 3 or 4 years ago?
Well, there's a longer story by which the CRISPR system was developed. It has its origins in the study of the immune system in bacteria.
Bacteria have an immune system?
Yeah, I mean, it's an amazing story, and I think it captures why so many of us are in biology in the first place. Researchers were studying how bacteria, like ourselves, bacteria have a system to protect themselves from viruses. They can be infected, much like we can. And the way that bacteria defend themselves is ingenious. It has a system that's very different from the way that our immune system works. Essentially, bacteria remember the viruses they've been exposed to. They take pieces of the DNA that come from those viruses and store them in their own chromosome. And then they use that information as like a hard drive, a memory of the viruses they've been exposed to. And part of the way that that memory is used to protect itself is this molecular scissors, protein called Cas9 that bacteria have, that human cells, higher animals do not have. And what this protein does, a protein that we've never had before in our arsenal to study biology, is a protein that can cut any piece of DNA, effectively any piece of DNA, by being programmed by this CRISPR locus, by this CRISPR-based hard drive. Effectively, it's like a molecular scissors that you can tell it what to cut in a very customized way.
So this was discovered in bacteria. How did it get to be applied to our cells and other animal cells?
Yeah, so over the span of many years, several biologists figured out how this system works. And they realized there's this molecular scissors as a key step of the immune system. And several scientists realized that if you could introduce these components into human cells, you could start to change DNA. You could start to cut DNA. You could start to paste in letters of DNA that we desire to put into the chromosome. And so going back only maybe 3 or 4 years, people have started to implement this. And it's basically swept through all fields of biology research. It can be applied essentially to any organism.
Give us an example of how you think it might change things. I think one of the examples you mentioned was the disease muscular dystrophy. How might CRISPR help solve that problem?
Yeah, so I think The applications are enormous. I think the thing that comes to mind for most of us are genetic diseases. I think we all know that mutations in genes are causing a large number of diseases. By and large, these are diseases that are among the hardest to treat because they have a genetic basis.
We're just baked into us, right?
Yeah, it's often inherited, and sometimes it's a single mistake in a letter of DNA that can be sufficient to cause a disease. And to date, we've never, in principle, we know how to fix the disease, switch that letter back to the right proper letter, but we've never had a technique that could do that efficiently enough to ever be applied to human beings. So these rare genetic diseases, rare or common genetic diseases, are fertile territory to now apply this approach and try to alleviate the suffering these diseases are associated with. I can give you one kind of example, but this is one of really countless that you could draw on. A disease like Duchenne's muscular dystrophy. We know the gene that's, when mutated, causes this particular disease. It's a very sad disease associated with weakening of muscles in children and adolescents. Ultimately, these patients succumb to death and inability to breathe using their respiratory muscles. We know the gene. We know the mistakes that can cause this particular disease. What scientists, this is not our work, but that of several independent laboratories, showed that you can use CRISPR, infuse via an injection, viruses that deliver the CRISPR machinery into muscle cells. The CRISPR machinery can go to that gene and change the DNA letters in a way that skips over the problematic mutation and basically allows this protein, it's kind of, you can think of it as a kind of spring that muscle cells use to deal with tension. It allows this protein to go from a non-functional, sort of short, ineffective version to a basically fully functioning spring, and allows muscles to work and deal with stress.
Now, we can't do this yet in humans, right?
This is quite a few years from a human clinical trial. This has been done in an animal that was genetically engineered to have these mutations. By and large, the field is at a moment right now where we are exploring in animal models how we might deliver these things and allow them to be efficient and to be safe. And I think these are the big unknowns at the moment of how safe and how efficient these will be to ever be given to a human being.
But by and large, genetic diseases, you think, might now be fixable?
I think a subset of them, the diseases in which CRISPR can be delivered, you have to deliver, remember these machines come from bacteria and have to be introduced into a human cell. And it also has to be something where you know enough about the disease to know the kind of change you want to make. A muscle disease also is free of some of the ethical dilemmas that might come up with CRISPR. The fact that you would change the letters of DNA in muscle, you wouldn't be changing it in the germline. And so these changes wouldn't be...
In the germline, you mean
So
it wouldn't be inheritable?
It wouldn't be inherited to the next generation. So that's a whole separate issue of when is it okay to fix a mutation in DNA? Well, most people would agree it'd be nice to fix the mutation in a human, but not have that go to the next generation because of a concern of unintended consequences.
We'll get back to that later on. So in this particular case, though, you can fix, in an animal, you can fix the gene and cure the disease in the animal. So one day, you might be able to do that in real people, right? What other examples of a disease that will be fixable that way?
Well, I think the diseases where CRISPR is most amenable are those in which you can take cells out of a human, genetically manipulate them in the laboratory, and then introduce them back into a human. So blood diseases are a real opportunity to apply this because you can take blood stem cells from the bone marrow and then do a bone marrow transplant. We have a lot of experience with applying bone marrow transplants in clinical medicine. We could now make an adaptation of that where we could take out a blood stem cell, fix a mutation, for example, in a disease like sickle cell disease, a disease where you have a defect in an oxygen-carrying protein. We've known the genetic basis for sickle disease for a very long time, but we've never been able to fix that mutation. In principle, if we work our way through all of these obstacles to setting this up, we could fix that mutation in a blood stem cell and then reinfuse those corrected cells back into that patient. And I think a lot of us are very excited and optimistic that blood diseases will be some of the first to fall, perhaps, with this approach.
Now, I know this isn't your field, but people are using CRISPR in agriculture, right, to improve plants, in insect control. In insect control, we were talking a little bit about the possibility of using this to wipe out mosquitoes.
Yeah, I mean, this gets a little bit more into science fiction, and I think this is very far from any real-world application. But there are ways, now that we have this technology, people can think of clever ways you could do something quite extreme, such as eliminate a problematic mosquito population by having a CRISPR, you could sort of think of it as a CRISPR chain reaction, where a genetic change made by CRISPR would sort of duplicate itself and self-perpetuate across a problematic species. Even as I'm saying it, it sounds a little scary as a concept, and I think most of us are not, no one is thinking seriously about that application. But in agriculture, it's already happening. There's a contentious issue around genetically modified organisms in crops. Taking foreign genes and introducing these into plants. I'm sure there are a lot of different opinions on whether this is the right or the wrong thing to do. CRISPR gives us an alternative to GMO where you could just inactivate naturally occurring genes into plants without introducing foreign DNA. And I think there are a lot of people that find that to be less contentious and potentially an alternative route to introducing foreign genes. So in agriculture, to make herbicide-resistant plants, to make plants with more desirable features to feed the world, I mean, there's an enormous excitement about this approach.
Now, your lab is specifically a cancer lab, right?
That's right.
How can CRISPR help the field of cancer?
So in several ways. I mean, first of all, using CRISPR as a therapy, it's very hard to think of doing that in cancer, unlike sickle cell disease, unlike muscular dystrophy, for many reasons. One is because cancer is a multi-gene disease. It's hard to think of fixing all the mutations that would cause a complicated disease like cancer. But there is a way we could use CRISPR therapeutically in cancer. There's a huge amount of excitement about using the immune system, now we're talking about the human immune system, to fight off cancer. You can take T cells, one type of white blood cell, out of a cancer patient. T cells in our body have a purpose of eliminating foreign organisms from our bodies. Could you trick a T cell into rejecting your own tumor?
Normally, it's not able to do that for some reason.
Tumors often are sneaky and find ways to sort of evade the immune system. But the idea is, could we sort of enter an arms race with the tumor where we take the T cells out, use CRISPR on the T cells,
to make them better T cells.
Yeah, like arm them properly, equip them to sort of then infuse those T cells back in to eliminate the tumor. Not with CRISPR, with a similar technology called CAR T cells. This is already being done. The ability to now use CRISPR as an additional tool is generating a huge amount of excitement. So that's CRISPR as cancer therapy. I think there's an opportunity. The second major way that CRISPR is having an impact in cancer is it's allowing us to look at the genetic changes that cause human tumors. And now, in a very facile way, we can introduce those same DNA changes in a mouse or in a rat.
Why would you want to do that?
And so we can make models of human cancer. And so if we want to test new therapies, we want to run kind of preclinical trials with new drugs, we can do this in a sort of humanized cancer model in a rodent, for example. And so the ability to make rodent models of cancer is exploding. It's become easier, cheaper than it ever was before. And then I would say the third application.
So let's just clarify. So you're saying you essentially give the rodent a human cancer, right?
That's right.
And then you can test out your therapy against that, and you hope then it would work.
Exactly. And this is not a new idea. We're already doing this, but historically, it's been a 5-year, 7-year project to make one cancer model. Just to give you an idea of the efficiency of CRISPR, I mean, this is something you can now do in the months time scale, what used to take years. And for us as scientists, training PhD students and trying to kind of move the ball down the field as aggressively as possible, you can imagine the efficiency of this just captures a lot of the imagination, particularly among young scientists entering the field.
Right. But then there's a third area, one that you're particularly interested in, right?
Yeah, exactly. So the main focus of my lab, the way that we are trying to make progress in cancer is to develop better, more modern drugs to fight cancer. To find things that are better than the toxic kind of chemotherapy agents that we use for most patients today. more targeted agents that could selectively disarm a cancer cell, but without widespread toxicity in the human body. We and many others are desperate to find these kind of Achilles heel vulnerabilities or weaknesses that cancer cells have that normal cells do not possess. Our research is fundamentally a search to find those types of vulnerabilities in cancer cells. CRISPR is the ultimate tool to explore in this area. We can now do things that we never thought were possible. We can now take a culture of cancer cells in a dish and now mutate thousands and thousands of different genes in a cancer cell, effectively probing through all the different proteins that a cancer cell possesses, and ask the question, which kind of proteins does a cancer cell need that normal cells do not require?
Wait a minute, so we all have proteins, they're the little machines that make the cells work, right? And you're saying that cancer cells have proteins that have gone bad, that are wrong, that enable it to be a cancer cell? Yeah. And those are the ones you're looking for?
Yeah, I mean, basically every cell in our body has thousands, tens of thousands, maybe 30,000+ different proteins. Proteins are basically little machines. We generally think of them as nutritional substances, but proteins is the machinery, one of several machineries that makes life possible, and they execute a variety of different tasks. Normal cells in our body have all these protein machines. Cancer cells have the same number of machines. Some of what causes cancer, the net effect of these DNA mutations, is to change and corrupt proteins in the cell. What we are exploring is which of these proteins does a cancer cell need to be a cancer? And which proteins does our normal biology need as well? And we believe that if we can compare and contrast the essential proteins in a cancer with that of normal, we could reveal these Achilles heel kind of weaknesses or vulnerabilities.
How does CRISPR enable you to do that?
So effectively, if what I just said is that DNA mutations, the ability to mutate a DNA effectively changes proteins. There's this beautiful relationship between the genetic code of DNA and the functionality of proteins. CRISPR allows us to change the letters of DNA, so in essence, we are perturbing proteins in a systematic, very efficient manner. And so we can look at a cancer cell. We know it has 30,000 different proteins. Well, let's perturb every single one of them.
Can you really do that?
It's
astonishing.
It's entirely feasible, and there are approaches, we call it multiplexing, which allows us to, rather than one scientist sort of setting up one experiment and testing one CRISPR experiment, in one experiment in our lab, a scientist and in many other labs can now manipulate thousands of different genes, make thousands and thousands of different mutations with the same amount of work. And so the efficiency of it is, it really opens up possibilities that didn't exist. And essentially, we are able to comb across all these different proteins. Proteins are very complicated, three-dimensional structures.
And their shape makes them do what they do, right?
Exactly. They have a very complicated function. A lot of what biologists, biochemists like ourselves do is study how proteins work. With CRISPR technology, we can now comb across the surface of these proteins, figure out not only which proteins does a cancer cell need, but which sort of surfaces on that protein does it need. And from the purpose of drug discovery, that is the kind of secret sauce of cancer drug target discovery. That is the fundamental objective of making new drugs in cancer. It's not just finding genetic weaknesses in cancer. It's not just finding essential proteins in a cancer. We want to know these little invaginations on the surface of a protein that we might stick drugs as kind of a lock into a key, a key into a lock that might allow us to disable a protein and make a cancer cell die.
Now, you already have something that does that, right, which you discovered pre-CRISPR. Tell us about that as an example of the sort of drug you're talking about.
Yeah, so in the pre-CRISPR era, so this was not nearly as efficient as how we do this now, we were able to apply a different technology to discover one of these kind of Achilles heel vulnerabilities in a disease called acute myeloid leukemia, which is a cancer of the blood, of white blood cells, actually blood cells that are normally part of your immune system. This is a very aggressive malignancy. It's one of the more common cancers that children get. It's a very devastating disease, and we've been hunting for quite a few years now to find all the weaknesses that we might be able to act upon in these leukemia cells that makes them different from normal blood. And we stumbled into one of these examples of a protein moreover, these specific invaginations on a protein that a leukemia cell...
Invaginations, little holes.
That's right, little pockets on the surface of these proteins that these leukemia cells desperately are attached to be a leukemia cell. Normal cells don't care about them so much. Entirely serendipitously, we had discovered that a chemist working totally independently of us in Boston named Jay Bradner, a good friend of mine, developed a small molecule targeting these exact pockets on this protein called BRD4. And through just total dumb luck and being in the right place at the right time, we were able to not only discover a vulnerability in leukemia, but act on it with a drug. And using these drugs, we could show that in an animal that has leukemia, we could extend the lifespan of these leukemia-bearing mice. 2 years later, several drug companies acted upon this discovery and started phase 1 clinical trials of very similar derivatives of these drugs designed for application in human.
To be able to get into the human and then fit into this little pocket in the
Exactly. It's basically a pill, orally available drug that goes into our cells and it sticks to these pockets on the surface of this particular protein that we identified. And we did a lot of work in animals. This is how we make therapies in medicine. We test it in an animal, make sure it's safe and effective, then you take it into human. And so 2 years after the discovery was published, it went into the clinical trials began by several independent companies. About 2 years later, we were sort of waiting to hear what the results were. Just this year, the first phase 1 trial was published. And what's been learned is, first of all, the initial goal of introducing a new drug into a human is asking, is it safe? And the answer is yes. So the doses were identified that patients can tolerate. The side effects are not so bad. And they're treating patients with acute myeloid leukemia, the disease that we were modeling in animals. And what we've seen is that a subset of the patients treated on this trial, these are patients that had failed all prior chemotherapies and standard of care, a subset of these patients have had responses to this drug, which hits, again, these pockets that we identified. Subset of these patients have had really quite dramatic responses, basically elimination of leukemia cells or partial elimination of leukemia cells. And so we've been very excited. The drug development industry has been very enthused by these initial results. It's very early days. It's all sort of small numbers of patients. We're still learning how to use these drugs, what to combine them with, how to make their effects more durable and longer lasting effects. But we've had basically this one-off kind of example of through kind of earlier genetics approaches, pre-CRISPR, had discovered a drug target. Again, I would say mostly through dumb luck, being in the right place at the right time, but seizing the moment. We're now very optimistic with this kind of enhanced genetic tool where we can now manipulate all the genes in a cancer cell and all the protein surfaces. We're trying to really take this same concept to an entirely new level of discovery. And so that's been our sort of angle on kind of embracing CRISPR.
Right. So finding the vulnerabilities of a whole bunch of different cancers, right?
Yeah, I mean, I think the dream discovery we are looking for, and we're finding them. They're hard to find. You need to do a lot of CRISPR experiments to find what we're actually looking for. But we want to find a pocket on a protein that a normal cell just does not need at all. So it's a protein that's in our body, but often normal biology is very robust. You often don't need every protein in every cell for normal biology. But occasionally a cancer, when it became a cancer and it sort of drifted off from normal biology, sometimes it becomes addicted to proteins that it didn't need before it was born. We're hunting by systematically perturbing every greasy pocket on these proteins, and we're looking for a perturbation that makes a cancer cell rip apart and die, makes it get eliminated from the organism. But with normal cells, just saying, who cares? And you can imagine, it's sort of the magic bullet of cancer therapy. You can imagine a lot of reasons why it's hard. But this is where CRISPR, again, is so advantageous, because we can basically, we don't have to bet all our chips on one kind of number. We can just not, we can hedge our bets by just changing everything. Do the largest possible experiment and find the thing that has that rare sort of magic bullet property. And it's early days. I have a lot of young scientists in my lab participating in this exercise. And we're finding things that were invisible to us with earlier forms of technology. And so remain optimistic that this is going to bring about new targeted therapies for cancer.
Must be a pretty exciting time in the lab.
It's a lot of fun. I mean, as an academic laboratory, we are powered by people in their 20s, come into Cold Spring Harbor and do a PhD. They take on a 5-year project. And you can imagine, this new technology is coming about. They can take on an ambitious project, try to discover a cure for a deadly malignancy. You can imagine
just in a 5-year time.
Yeah, I mean, I think it opens up the imagination to explore and find things that wouldn't have been possible. And so a huge part of my job is to excite and encourage and train the next generation of scientists. This is part of our mission in academia. And a revolution like the CRISPR revolution only makes my job easier.
Now, we have to address the issue that some people are nervous about, right? That you could actually start changing genes in making people better one day in the future, or literally changing how humans are, changing the basic genetics of people. Is that likely to be possible with CRISPR?
Possible, absolutely. I think everything, if you look at the trajectory, the pace with which discovery is being made, it's going to be possible. The more relevant question is, should we ever consider doing this? The diseases I just mentioned, where you manipulate DNA in your blood or in your muscle, and it doesn't go to the next generation, won't go on to your children, I think most of us would consider that's something we should explore. Well, there are diseases that do transmit from generation to generation. If you could fix that mutation and alleviate the suffering once and for all for you and your offspring, I mean, this is a debate, this is a discussion that we all need to participate in, not just scientists. It's beyond sort of our ability to judge at this point. I can say that by and large in the scientific biology research field, no one is advocating to go in and do this. I think almost everyone would agree that we should be very patient and we should explore the capabilities of this technology. Let's see how safe it is. Let's see what it's all about. See what the unintended consequences are in animals or in a less dangerous sort of experimental system. That's where we are today. I don't think anyone is advocating to fix mutations. No one's advocating for enhancements of traits in the, I mean, this is still, you might see it on the cover of a magazine. There's a dialogue that we should appropriately be having. But this is something that I think most scientists would agree that we should all be participating in as a society, as a community. And
yes, but because it's so simple to use, right? Anybody can start doing this. Somebody that maybe not in the mainstream scientific community, who's not part of this more cautious approach. How do you, the rogue experiment somewhere, how are you going to prevent that?
Yeah, I mean, I think...
And already there's concern that scientists in China have been manipulating human embryos, not viable ones, not in any way getting to what you were just describing, but it just shows that people are beginning to think about that. And how do you control it outside the mainstream of science?
Yeah, it's obviously a challenge to control these things. But it's also not, you have to keep in mind, this is also nothing new. There's been a recombinant DNA technology that's emerged in the 70s. The idea of human cloning, I mean, this is something we've been discussing and have been regulating for some time, working with embryonic stem cells. There are measures that can be taken at a policy level to control the access of the technology and how this is implemented. But you're right in the sense that by virtue of things becoming easier, there has to be a reciprocal discussion about how things are regulated to make sure this is not done in an ethically dangerous manner. And so there have been large consortia to kind of discuss this challenge head on that's happening at present in the community.
Do you think CRISPR will get the Nobel Prize?
It absolutely should. Who will get it is a tougher question to answer.
Why is that?
Because I think the Nobel Prize is an awesome thing. It inspires a lot of people to think about big accomplishments in science. But the reality is a lot of discoveries in science happen not by one person or by 2 people or 3 people. It's by many scientists over the span of many years.
And only 3 people can get a Nobel Prize.
That's right. So I mean, I think it's in some ways, especially in modern biology research, it's more and more sort of a team sport. And in some ways, it's maybe we need to rethink how we give out these kinds of prizes to acknowledge the fact that no one works in their garage to make these discoveries on their own. It's large teams sometimes, and in the case of CRISPR, it's many people that have contributed to this coming out of the bacteria immune system world on into human applications. And I think there's a lot of people that share the credit for bringing this about.
But right now, there's a sort of fight going on, right, between there are two rival institutions claiming priority in this field. One is in Cambridge, Massachusetts, and the other is in Berkeley. And this is I think one of the reasons why we didn't have a Nobel Prize this year, because the committee didn't want to crown any one of these individuals. It's a multi-billion-dollar debate, right?
That's right. I mean, I think it's important to also recognize that out of this academic, very basic research has been a huge rush of biotech companies, pharmaceutical companies that have embraced this to develop new drugs, new products, and this has sort of energized medical research also in the private sector. And so
there are companies now that
there are many companies that have started with the goal of bringing CRISPR to the clinic. Right. And so it's happening at all levels. And so naturally, it makes some of the intellectual property and the patents quite lucrative, which is the subject of continuing debate.
Right. Well, you don't have to worry about that, right?
Yeah,
you just get to use it.
I'm very proud to say I had nothing to do with the discovery of CRISPR, but I'm a proud user of the technology for the kind of cancer biology research we do.
Well, Chris, thanks so much for coming along. I really appreciate it. Let's hope that CRISPR leads to lots of good things in your lab and others as well. Thanks so much for coming tonight.
Thank you. Cheers.
Thank you. And thank you to Stony Brook Yacht Club. Good night.